Okay, for problem 719, they're actually giving you the map position for three different genes on chromosome 6 in maize or in corn. Um, Here we've drawn them out. This is what they give you in the problem. The three different genes. One of the genes is coding for leaf color, purple or green, as you can see, I designated it there up there. For the silk color, yellow or salmon, and for just the regular, just the size, either normal size or pygmy. They're all located on the same chromosome at these different positions here. And so you know that there's 10 centimorgans in between the PL and SM, and there's 10 centimorgans between SM and PY. And so here they're just sort of getting you to kind of do exactly opposite of what you've been doing up until now. They give you the results for the crosses, and then you have to calculate those distances. Now they're giving you the distances, and they're saying, okay, in this cross, in the offspring, how much, in order to get these distances, what should you have seen, right? Okay, so here I've just drawn out the initial cross. So it's heterozygous, or it's homozygous for the dominant, the recessive, and the recessive, and then homozygous for the recessive, dominant, right? And so when you get the cross between that, it's heterozygous in each of those genes. But because it was set up this way, we know that on one chromosome you have a big PL, little SM, little PY, a little PL, big SM, big PY. Okay, so now you know the phase of the alleles on each of the chromosomes, and then so in this way we'll be able to tell or predict sort of the crossing over events and be able to see them, right? And so you're now crossing with a recessive male, and it's saying, okay, in the situation where there's no interference, what is the percentage of each of the phenotypic classes would you expect? And so you're basically going to have to reconstruct the phenotypic classes that would give you this 10 centimorgans, or 10% of the time you're getting the crossing over event occurring between these two genes, or the 10 centimorgans here where you'd have the crossing over events occurring, in 10% of the gametes that were produced. Okay, and so with no interference, that means that your expected number of double in the phenotypic class where the results from double crossing over is the exact same as the ones that you observe, right? And so because there's no interference going on, you would say, okay, what in the creation of a gamete here, this gamete, right? The gametes that were produced from this individual, and if you had what percent of the offspring would you have expected a double crossing over event to occur? And so here we need to go back and think about, okay, what do 10 centimorgans mean? It also means that in 10% of the time, or there's a 10% probability that you're going to get a crossing over event that occurs between SM and PY, and then there's a 10% possibility that there's going to be a crossing over event that occurs between PL and SM. Okay, so if these are two different events and each of them are 10% or 0.1 probability, the probability that both of those are happening is then 1%. 0.1 times 0.1 equals 1%. So here you come over to here and you say, okay, so if there's no interference, then we should expect the double crossing over events to occur in 1% of the offspring. Okay, and so we see that here are the two different genotypes from this cross, in the offspring from this cross, that would result from that double crossing over event. And so you say, okay, there's going to be a total number, 1%, and so then a half of 1% and a half of 1% would have to come from each of those phenotypes. So now we have the double crossing over events, okay? So now we can go to the single crossing over events. So let's just go down here to what I've designated CO2, crossover 2. So between SM and PY. And so in order to eventually get this 10% here, or these 10 centimorgans here, that means that 10% of the phenotypes have to have had a crossing over event that occurred between these two genes. So you already see here that in 1% of the phenotype, you've had that crossing over event that occurred between these two. So then in the second, or under the CO2, that has to, because the total amount has to equal 10%, you therefore can assume that there has to be 9% here in order to add those two up to get the 10% that would have resulted in calculating 10 centimorgans distance between these two genes. Again, there's two different phenotypes, and you can actually go through and do this crossing over event, and flip it and say, okay, that's what these phenotypes would look like. These two phenotypes came from a crossing over event here. Here's the phenotypes, here's the genotypes of it. But again, they have to add up to 9%. And so you have 4.5% of each of these phenotypic classes that would contribute to that. Okay, same thing with CO1, right? Again, you're going to have 9% of the phenotypes come from that show that there's a crossing over event occurring between PL and SM, because you've had 1% here, right? And when you add all those up, you then get that 10, which would have given you your 10 centimorgans. Okay, so now we would have 6 of the 8 phenotypic classes accounted for, 4.5, 4.5, 4.5, 4.5, 0.5%, and 0.5%. Therefore, the rest of them, it has to sum to 100%. The rest of them that you just minus 100 from all of these other ones, and you get 81%, where there was no crossing over event that would occur. To check if you've done that right, you could just go back through and give some numbers to all of these, right? And then see if you calculate 10 centimorgans between each of those genes. Okay, so that's no interference. Now let's look at when there's a complete interference. So what complete interference means is that there's going to be no double crossing over event occur. So here I've just, if there's a crossing over event in one, it makes it so that there's no crossing over event occurs between the other two genes. And so you'll have 0 double crossing over events. And so therefore, if you were to calculate the 10 centimorgans, you would have had to have 10% of the offspring that showed the crossing over event that occurs between what was the gene SM and PY, and 10% here that would have to occur between the other two genes. Therefore, the rest of your phenotype, or your rest of your phenotypic classes would have to add to 100. So then it would be 80% here, and so that's where you'd have those adjustments. You'd have to have those percentages in order to calculate 10 centimorgans between those genes. You could then go back, and I've numbered those different phenotypic classes that are the same as what they were over here. And you'd then just have 5% here, 5% here, 5% here, 5% here, and 40% and 40%. Okay, so that's that problem.